to you all, and thank you for dialing in tonight to the next episode of the 2023 State of Play series. For those of you on Zoom, may see a notification about the meeting being recorded. Please just click OK or agree to continue. First up, an acknowledgement of country. So in the spirit of reconciliation, MND Australia acknowledges the traditional custodians of country throughout Australia and the connections to land, sea and community. We pay our respects to the elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. And I'd like to acknowledge the Wiradjuri people on whose land I am and pay my respects to elders past, present and emerging, and extend a welcome to any Aboriginal and Torres Strait Islanders peoples on this call today. So today's theme is around investigating the motor neuron muscle interface in MND, which obviously plays a critical role in many of the most disabling aspects of the disease. We're fortunate to have two leading researchers in this area presenting tonight, both who were funded this year by MNDRA. We have Professor Aaron Russell from Deakin University in Victoria and Associate Professor Peter Noakes is at the University of Queensland. As previously, each presenter will talk for about 20 minutes and then we'll have a combined Q&A session at the end. If you have any questions, please submit them through the chat or Q&A function if you're on Zoom, or if you're on through the comments if you're watching this through Facebook. So that's all you need to hear from me. So Peter, please take it away, share your screen and it's all yours.
Can you see my screen?
Not yet.
Can you see it now? Can you see it now?
Yep, perfect.
Okay, good evening, everyone. Thank you very much for MNDRA for inviting me to give a talk. And so I'm going to talk about the motor nerve muscle connections and what we've been doing in that space, and then that'll feed into my colleague, Professor Aaron Russell, who will actually take you inside the muscle and talk about muscle in a bit more detail. So why would we want to look to target muscle to assist in the treatment of motor neuron disease? And the reason is we normally think that the fault really lies with upper and lower motor neurons and the surrounding cells in the brain and spinal cord. So when we look at the motor nervous system, we have, and this is my little cartoon here, we have the brain, we have the spinal cord, and in the brain we have upper motor neurons and motor neurons located in the brainstem just below the brain that connect with lower motor neurons. These are called alpha motor neurons. And these alpha motor neurons will send their projections outside to the periphery to connect with skeletal muscle that allows you to move. And so we talk about this as a motor nerve muscle connection. Currently, there are treatments being developed that really target the brain and the spinal cord in controlling inflammation within the brain and spinal cord. But there are very few treatments that are sort of working out in the periphery where the motor nerve makes connections with its skeletal muscle. In this little box over here is actually a human neuromuscular connection. And so what you see here stained in red is the motor nerves coming down. It branches into two branches and makes a connection on the surface of the human muscle cell. And in green are sort of the specialized receptors that are in the muscle membrane that allow the motor nerve to talk to the muscle to allow it to contract. We typically think of these endings as little grate-like endings of the nerve endings that sort of sit on top of the muscle. And so we've been working for many years at this level, understanding what's going on between motor nerves and muscle. So what I'll cover in my talk today is evidence from our lab that motor nerve muscle signaling is important in keeping motor nerves connected to muscle, particularly in the early stages of motor neuron disease. And our study has been done with the kind donation of muscle samples from MND patients and their families. So this work is really dedicated to you and to all the people that are interested in motor neuron disease. And we've discovered some faults in motor neuron disease muscle, which include faults in motor nerve to muscle signaling. And then towards the end of my talk, I'll talk about how we might go about correcting some of these faults in muscles with the idea that we may be able to improve muscle strength. So I don't need to tell anyone what motor neuron disease is, but it's obviously a very tragic disease. But it's best characterized in the clinic as an early loss of motor nerve muscle connections, and that manifests in muscle weakness and eventual paralysis. We all know that it involves the loss of upper and lower motor neurons within the brain and spinal cord. We've been working for a number of years looking at the loss of motor nerve muscle connections is due to a fault in signaling between motor nerves and muscle. This is something we've been working on for many years, not only in normal development, but obviously in motor neuron disease. And we believe that muscle may have a role in this loss of motor nerve muscle connections. And so what evidence do we have for this belief? Well, if we look at normal nerve muscle connections in healthy patients and in motor neuron disease patients, we notice a number of things. So here we have the motor nerve coming down and it forms this ending on the muscle surrounded by these green, what we call acetylcholine receptors, these muscle specializations. And it looks like this grape ending and it's quietly, tightly compact. And here's another example here. By contrast, when we look at the nerve muscle connections in motor neuron disease patients during the early stages of the disease, we see a lot of the nerves thinning and starting to move off the contacts with muscle. We also see that the specialized region, which is in green, these receptors that are embedded in the muscles, starting to disperse and disappear away from the muscle. And so we think of these sort of changes that we see at this sort of observational level as a sign that we're losing communication between motor nerves and muscle. And so my colleagues and I have sort of said, well, how do we go about investigating this? Beyond looking at the muscle biopsies from MND patients and comparing them to normal patients, how might we go about looking at this? So about several years ago, we got together with the clinicians at the Royal Brisbane and Women's Hospital, and we set up a system where we could take a muscle biopsy from a patient and look at their what their nerve muscle connections look like, which I've just talked about. But more importantly, we've developed methods where we can take these small biopsies that we collect from patients and their family members, and we can grow them in tissue culture, in a dish. And we can actually form muscle cells in a dish, which we can use to study. And over the last few years, we've been able to basically put these muscles in one compartment of a dish, just like a compartment out in the limb. And in another compartment in a dish, which is separated, we can put our human motor neurons in a culture, and these axons will be sent out to the muscle chamber. So this would be a little bit like the spinal cord sending their motor nerves out through the arms to connect with muscles in the limbs, for example. And then when we look at how the motor axons, the motor nerves, run over the surface of the muscle fibers, which is here indicated in blue, we start to see the formation of these large receptors in the muscle membrane, these acetylcholine receptors that I'll talk about, similar to what we're seeing here in the real muscle here, the accumulation of these receptors in the muscle membrane. So when the nerve contacts the surface of the muscle, it induces these sort of muscle specializations. So taking this sort of assay where we've developed a motor nerve muscle connection in a dish, we can start to study and compare normal muscle to muscle that we can get from MND patients. And what we've been able to show is that nerve muscle-induced specializations, these clustering of acetylcholine receptors, while they occur in a normal muscle when the motor nerve grows on top of them, when the normal motor nerve grows on top of a muscle from MND patients, we don't get this big clustering of these patches of muscle specializations, these acetylcholine receptor patches. So we've done this for about 10 or 11 patients. Some carry mutations to motor neuron disease, but the majority are sporadic. So we don't know what the genes are that cause disease in those cases. So we see this as our simplest explanation for this is that the muscle is failing to respond to the presence of motor nerves. So what could be wrong? Well, one thing we know about nerve-muscle interactions during normal development is that motor nerves actually release proteins that can induce muscle specialization underneath the motor nerve ending when it contacts muscle. And these specializations are needed for muscles to contract, which gives you muscle strength. And one of these proteins is called agrin. And so agrin is made by motor nerves. It can be released from the motor nerve, and it binds to this sort of receptor complex, which is embedded in the muscle membrane directly underneath the motor nerve. So it binds to this LRP4, this MuSK, DOC, and this activation of these receptors will cause the acetylcholine receptors, this muscle specialization, to occur in the muscle membrane directly underneath the motor nerve, so that when the motor nerve is stimulated, it can release acetylcholine, bind to these receptors, and cause the muscle to contract. If you don't get clustering of these receptors, then you have muscle weakness. And we know that from a variety of other neuromuscular diseases such as myasthenia gravis. So what we wanted to know was if MND muscle doesn't sort of cluster these receptors, could it have something to do with this pathway? In other words, how do muscle cells derived from healthy controls and MND patients respond to agrin treatment? So we can grow our muscle cells up in a dish, and we can grow control muscle cells, and we can control muscle cells from MND patients. And we can add this protein that is made by the motor neuron, and we can add it into the cultures of the control muscles and of muscles from MND patients. When we add it to control muscles, we get the clustering of acetylcholine receptors in the muscle membrane, this green stuff, just like we see the clustering of acetylcholine receptors in the muscle underneath the motor nerve. By contrast, when we add the agrin onto the muscles from MND patients, we get these little spots of acetylcholine receptors, these patches, but we don't get big ones that we would see under normal conditions. And so this is interesting. This tells us that maybe in the MND muscles, this sort of pathway that agrin can use to cluster these patches into large patches of acetylcholine receptors may be faulty. So we've been looking at this sort of in a bit more detail in terms of what is wrong with the muscle from MND patients. So this looks a bit weird, but basically what it is a protein blot. So we can grind up the muscle from control, and we can grind up the muscle from MND patients, and we can run it out, and we can probe for specific proteins that are part of the agrin pathway to cluster these acetylcholine receptors in muscle. So what we know in control is that we have a lot of DOC7, but in MND patients, we have very little of it. And that is just graphed there. In control patients, we have a lot of LRP4, but by contrast in MND patients, we have a lot less. So this tells us that this molecule and this molecule is reduced in the muscles from MND patients. So if these molecules are reduced, maybe when agrin's released, it can't effectively signal through this complex to cluster receptors. That's one interpretation. And so with this in mind, can we target muscle to develop treatments? In other words, can we add some more LRP4 back up to levels of control? And can we add some more DOC7 up to the same levels that we see in control muscle? And adding extra DOC7 or LRP4, could that make the muscles from MND patients responsive to agrin to cluster receptors? And in so doing may help return normal motor nerve muscle function. In other words, the title of this webinar is, can we target muscle to develop treatments for MND? So we've been working on adding additional DOC7 or additional LRP4 to stabilize the loss of motor nerve muscle connections to maintain muscle strength. What we already know is that the people in Japan and some people in New York, some colleagues of mine, have already added additional DOX7 into an MND model mouse, this SOD mouse, which you may have heard from various researchers before. And when they did that, they slowed the loss of motor nerve muscle connections, they made bigger nerve muscle connections, and the mice were actually able to live longer. But this is a long way between a mouse and a human. So there's a lot more work to do. I'll talk about what we're doing in that space in a moment. For LRP4, we've just been given funding from MNDRA to basically take a mouse that overexpresses LRP4 in their muscles and cross them to the SOD animal, this motor neuron disease animal model, and see if we can, by having additional LRP4 in the mouse muscle of the MND model mice, we get the same result as for DOX7. In other words, the mice have better muscle contractions, they move better, and they live longer. We do know that if you lose LRP4 in muscle, that you get a loss of motor nerve muscle connection breaks down. So we already know that. So we're thinking that if we can give additional LRP4 or DOX7, we may be able to rescue the phenotype. In other words, rescue this poor response to agrin by the MND muscle. So when it comes to human muscle approaches, we are working on two strategies. So obviously we can't go straight into the human and do all this. This has serious ethical problems. But what we are doing is we're taking advantage of the donations from the MND community and their family and friends to grow our muscle cells up in cultures, just like I've shown you, and infect them with non-infectious adenoviral vectors that can deliver additional DOX7, for example. This approach is actually quite an exciting approach because this is a sort of approach that's been used to rescue the dystrophin gene in boys that suffer from muscle dystrophy, and that's undergoing some forms of clinical trials. So this is a very interesting approach. The other approach is to use lentiviral vectors. And the reason why we need to do this is because LRP4 is a much bigger gene than DOX7, so it doesn't fit into these adenoviral vectors. So in the next slide, I'll show you some very preliminary data that we've got that allows us to go from optimizing delivery of DOX7 into muscles in culture to be able to doing it into human muscle cells. So these are mouse muscle cells. And so when we basically have given a DOX7, it can enhance agrin's ability to make huge clusters. And what I mean by that is that we've just got a dish here with just some muscle cells. Nothing's been added to it. And there's very few of these red acetylcholine receptor patches, these muscle specializations. When we add agrin on, of course, we get bigger patches in a normal muscle because this pathway is working to cluster receptors. If we give just additional DOX7 by an AAV adenovirus, and this is in green, so all the muscles are expressing the virus, and the receptors, we get some bigger receptors, bigger than what we see under adding nothing. But if we add the DOX7 plus the agrin, we get these huge clusters of receptors. And so now we know that in this system where we've given additional DOX7, that we've amplified this whole signal to a point where we've made these really, really big clusters. And this is in control muscle. So we're really excited now to be able to say, okay, this system has been optimized in a mouse muscle cell line. We now want to take our primary muscle cells that we've been given from the donors, the MND and their control patients, and we want to add it to our human muscle cells. So that's something we hope to be doing over the next few months. We do know, as I said, if you give DOX7 therapy into motor neuron disease model mice, that they get these huge neuromuscular junctions. So this is a normal mouse. These are these receptor specializations that are here in the muscle in a normal mouse. In an MND mouse, then these receptors break down. And this is a sign that the nerve muscle connections are breaking down. When they give a DOX7 in the MND mice, these individual motor nerve muscle connections almost double in size. And so we know that this pathway is very effective in the intact animal. So the hope is that we can see this in in the culture of the MND human muscles that may open some future therapies that we may explore in coming years. So that's our next step. Can we target human muscle cells with an AAV DOC7 non-infectious adenovirus? So I'm coming to the end of my talk, and really what I've shown you today is that MND muscle cells do not respond to the presence of motor axons. So here's some motor axons. Here are these big receptors of acetylcholine that sit in the muscle membrane that need for their muscle to contract. In MND muscle, this is from another patient, we don't get these big clusters. If we add agrenon to control muscle, we get big patches of these acetylcholine receptors. But if we add agrenon to MND muscle, we don't get them. And we've done this, as I said, for 11 patients. One of them has a SOD mutation. We haven't done it for muscles that are carrying the MND mutation, or but we have done it for the chromosome 9 open reading frame 72 mutation, and we see the same thing. So we see for both sporadic and familial. We think this non-responsive of MND muscle could be due to losing LRP4 and or losing DOC7. But if we lose both, then we really have lost this pathway's ability, if agrin's being released from the motor nerve, to cluster these receptors underneath the motor nerve to allow the motor nerve to stimulate the muscle to contract. And finally, I've shown you that at least we've been able to optimize delivering a DOC7 AAV into muscle. And when we add agrenon to it, we get these big clusters similar to what people have done when they've done it in a mouse model. Our next step is to do this into the human to see if we get a similar result. We also want to be able to do this for LRP4, but as I said, we're using a lentivirus, which we've just made, and hopefully by the end of the year, we should have some exciting results for that. So this work is dedicated to the MND patients, friends, families, and donors, because this is a human study. None of this would have happened without the donation from the MND patients, the friends, families, and donors. It involved people in my lab. It's involved collaborating with some of my ex-students, you know, people from the collaborators that have helped us make human motor neurons from stem cells that we can co-culture. Our clinicians, Rob, Pam, who run the motor neuron disease clinic at the Royal Brisbane and Women's Hospital, Lindy Jeffrey, who's a and Michael, who are neurosurgeons at Royal Brisbane, who helped us collect some surgical samples, which was very important for us to be able to look at the NMJs. And I've got questions here, but as I said, I'll leave the questions to after Aaron's talk. So I'm going to introduce Professor Aaron Russell from Deakin University. Aaron is going to talk about other aspects of the muscle, which are really, really exciting and may have real consequences for maybe how the muscle talks back to the motor nerve. So while we've discovered some molecules in the agrin pathway, there is a lot of other things going on in muscle that Aaron's now going to talk about. And I'll leave it there.
Thanks, Peter. You stopped? Yeah, you stopped sharing your screen. Okay. You see, is my screen now on presentation mode, or is it?
No, you're still on the slide mode, Aaron.
Okay, I'll go back again. As we said, you do this almost 1,000 times, you still can't get it right.
Oh, never worked the first time.
Okay.
It looks like it on it. You're still on.
Oh, okay.
Still on slide mode.
I'll try to get up. Sorry about that. I'll go again. Okay. It's not going. Sorry, it's not going to slide mode to presentation mode. Worked before. Sorry about this. Peter, you've stopped sharing your screen, yeah?
Yep.
Yep, okay, sorry.
Just go to the slide mode. It should be fine. I think it's pretty big in the middle there.
There we go. Have you got it now?
Yeah,
Yep.
perfect. There you go.
Okay, thank you. Sorry about that, everyone. Again, thank you very much for the opportunity to speak tonight. Personally, I find it's a really exciting area of research, the fact that people are now becoming more and more interested in skeletal muscle as potentially a major contributor to MND onset and progression. And again, thanks everyone for giving up their time this evening. As I'm going to mention that this is really a novel area. People have been dabbling in the area of skeletal muscle for a few years, but it's mostly been focused on the brain and spinal cord, as Peter mentioned previously. But the interest in skeletal muscle as a major actor in the disease comes from some interesting results that have been observed with respect to major factors embedded in the muscle itself. That's important for maintaining muscle health. And some of these factors are also very important for maintaining the connectivity between muscle and the nerve. And Peter spoke a lot and showed some very elegant images on the neuromuscular junction. That key organelle there that is the major link between signals coming from the brain through the spinal cord and that connecting to the muscle. And those molecules that Peter mentioned that sit at the nexus there between the muscle and the spinal cord are key aspects of maintaining signals. But there are also other factors further down within the muscle that could be contributing to the loss of the connectivity. So in my talk today, I'd like to, I'm assuming there's not a large number of muscle physiologists in the audience, and if that's not the case, I apologise in advance. But I'm really keen to provide some of the fundamental information around why skeletal muscle needs to be considered as a key player in MND. Several of these processes that I'm mentioning are important because they have been identified to be perturbed in muscle from people with MND and mouse models of MND. I'll explain why they're important to muscle health. I'll also explain how some of these processes may impact the connectivity in the MND condition, and also give you a brief overview of what my team is doing at the moment, very, very early on in a new program, to try and understand how these factors within the muscle may be affecting MND progression, and to determine if they are actually do have therapeutic potential. So the overlap with Peter's presentation will be referring back to the neuromuscular junction and also potential use of AAVs, as Peter alluded to early on. So I'm going to touch on a couple of key factors that are important in muscle health. Again, this is not an exhaustive list, and I hope I can narrate a nice little story around how these things interact together in the muscle and to protect the nodes here that Peter had spoken about, the neuromuscular junction, and how these impairments affect the skeletal muscle and then feed back up from the muscle here to cause neuromuscular degeneration. So a couple of aspects in skeletal muscle that we need to consider in the area of MND, and that's that there's a large amount of muscle fibre atrophy. So the skeletal muscles, some of the skeletal muscles become really small. There's breakdown of the proteins that cause the muscles to contract, and within that, the connectivity between the muscle fibres here and the neuromuscular junction occurs. But there's a couple of aspects within the muscle that's important not only for muscle health, but also for health of the connectivity between the neuromuscular junction and the tissue itself. There's the mitochondria that plays an important part. There's the activation of satellite cells that plays an important part in healthy muscle. And there's also aggregation of certain proteins that play an important role in their shuttling from one area to another. However, in an unhealthy situation, the regulation of the mitochondria and the satellite cells and the unwanted aggregation of certain proteins may impact their ability to keep the muscle healthy and in their ability to keep the connection between the neuromuscular junction and the muscle. Because these factors here, the mitochondria, satellite cells, and this TDP-43 protein, are important in maintaining the connectivity between the neuromuscular junction and the muscle. And again, our understanding of the perturbation of these three examples here do come from a couple of mouse models of MND and also from muscle samples that have been donated from people with MND. I'll touch briefly on this TDP-43 protein because historically it's been very well studied in the brain and spinal cord, where it aggregates in parts of the cell where it shouldn't be. However, we've identified, a couple of studies have identified its aggregation in muscle samples from people with MND, and we've recently observed it in the muscle of a mouse model of MND, where in theory it should only be in the brain and the spinal cord, but we are finding it in the muscle. Before I jump more into the molecular side of things, I'd like to give you an analogy because some of these molecules can be a bit confusing, but in fact, you can make it quite simple. And I look at maintaining the health of the muscle like maintaining the health of the car, in that we want to have a healthy car to move us around. How fast the car goes and how far it can go will depend on the engine of the car. It's also going to be affected by the fuel that we use to put in the car. You need the right type of fuel. Like our muscle car, every now and then there might be a pole in a car park that doesn't move, but it might, and you bumps into your car or you bump into that. And you're going to need someone to repair a little bit of damage to the car. Also, you're going to have a regular health checkup of the car where multiple parts of the car are going to be looked at in order to make sure that it's working optimally. And you can use these examples here to reflect on how those key parameters that I spoke about are actually important for maintaining, firstly, the health of the muscle, but also maintaining the healthy connection between the muscle and the nerve. So if I take this analogy here back to where I was speaking about before, our muscles, like a car, are important to move us around. They need to be healthy. If we have the disconnect between the brain, the spinal cord, and the muscle, we're unable to move properly. And that's obviously the case with MND. Within the muscle, it's made up of really important components referred to as the motor unit. And that's the muscle fibers and the neuromuscular junction. Now, like the car's engine, we can have different types of motor units. The car, you can have a V6, 4-cylinder, fuel injected, sports versions or whatever, in a particular muscle. So in every one online, we'll have a motor unit that might work like a V6, 4-cylinder. We might have a motor unit that functions slower or faster than others. Within these motor units, you've got the mitochondria. This is to produce the energy that's required to fuel the muscle to contract that allows us to move, such as your fuel is important for the car for you to drive the car. The energy produced by the mitochondria is also important to work in conjunction with these satellite cells, in case you've got a little bit of damage in your muscle. You sprain a quadricep, you sprain a calf, most of it have done some minor injury at some stage. And that gets repaired naturally when these processes are working well. Also, when this TDP-43 protein is able to move from one compartment, this little spot here, into a fibre, that helps with the repair, but then it goes back to its main spot, as you see with the fluorescence here. The mitochondria and the satellite cell and the TDP-43 protein is not only important for maintaining healthy muscle, but it's also important for maintaining the connection between the nerve, neuromuscular junction, and the muscle itself, because they do reside in proximity to the attachments that Peter had spoken about. And if there's a perturbation in the mitochondria, its ability to produce the right amount of energy at the right time, and if our satellite cells aren't able to become active when we require some form of repair, we may lose connectivity between the neuromuscular junction, or the signals that are coming from the brain via the neuromuscular junction to the muscle. Additionally, when maintaining the overall health of the muscle, and this is one protein that does that in a very elegant way. It maintains numerous processes within multiple tissues, but especially in skeletal muscle. When it's doing its job, one of its jobs, it remains inside the nucleus, which is found here in these little green spots. However, occasionally it needs to exit and forms diffuse clusters here within the muscle fibers. In a healthy situation, when the job's done, when the servicing's been done in your muscle, the diffuse aspect here of the TDP-43 reverts back to where it normally resides in the nucleus, and you won't have it sitting here in the muscle fiber. When all this happens, everything's happy. However, when it doesn't happen, when the mitochondria is not functioning right, when satellite cells aren't functioning right, when TDP decides to stay in a spot it shouldn't be for far too long, we end up with a disconnect at the neuromuscular junction. And by that occurring, the signals are not being received by the muscle from the neuron, and also the muscle is not able to recommunicate back up. So the question's often been asked, does the disconnect here occur from signals coming down? Or, as Peter and I are interested in, are there signals that are coming from distally from the muscle that are causing the disconnect to occur? I'd like to focus now on the engine of the muscle, the motor unit, because studies in people with MND and in mouse models have indicated to us that certain motor units within the muscle, so you have different types of motor units, may give us a clue to some of the molecules that could be negatively impacting, that may be causing this disconnect. But there also might be parts of certain motor units that are actually protective. And if we understand what they are, then manipulating those protective motor units, the protective environment, may be a strategy to delay the progression and the severity. So as I said before, you can get different types of powerful engines in cars. You can get different types of powerful or less powerful motor units within a muscle. Some of you may not have heard, we have different types of muscle fibers and muscle environment. We have a slow muscle environment. So all of those people who have enjoyed doing endurance type sports will have mostly this slow environment. And it's characterized by smaller, thinner fibers with smaller innervations through the NMJ. The other end of the spectrum, you'll have a fast, more powerful motor unit, more like a V6 engine. And that produces much more force very rapidly, but for a limited amount of time. Looking at the muscle and looking at this environment, we see that the slow muscle environment is impacted very late in the disease, suggesting it may have protective properties. While looking at the fast motor unit, they're actually impacted very, very early and often impacted before many clinical symptoms are observed. This has led us to consider the differences in the slow motor unit environment compared to the fast motor unit environment. And we've asked the question, if we're able to promote or enrich the slow motor unit environment, are we able to slow down or delay the progression? There's been some nice work done in healthy animals where researchers in Sweden have identified a particular protein referred to as neuturin, which has been studied extensively in neurons, not in muscle. And neuturin is known to improve the health and the stability of neurons. But only recently has it been identified and considered as an important factor in the maintenance of muscle health. And indeed, in healthy mouse muscle, when we increase the amount of this protein, there's a very clear observation that these mice create a slow motor unit phenotype. And I'll show you some data from previously published work to support this. So for example, now please remember this is I'll be comparing healthy animals with healthy animals that have more of this particular protein. So this is a neuromuscular junction, as Peter had identified, I showed you previously. And a slower neuromuscular junction is more tightly compacted with smaller nerve endings here compared to a faster form. So this is one indication that the NMJs are mimicking or appearing to be have a slower innervation. In a slow muscle phenotype, you have more of those slow fatigue-resistant fibres. And with this particular stain here in an oxidative or slow muscle, you see the mice that overexpress this neuturin protein have much more slow fibres, as indicated by the blue dots here, relative to the normal mice. Additionally, when we look at a typically fast muscle, the number of red spot, the amount of red here, and you can see in this blown up image, these red dots have been stained for the fast muscle fibres. And you'll see in the mice that express more of this protein, they have less of the fast, meaning they've got more of the slow. So in a slow muscle and a fast muscle, when overexpressing this particular protein, we're able to move or shift the phenotype to a slow oxidative phenotype. With respect to the mitochondria, as I mentioned before, the main energy producing organelles within the muscle, that's important for muscle contraction, that's important for muscle continued contraction, important for the neuromuscular junction stability. The mice that have more of the neuturin have more mitochondria, as indicated by the greater amount of darker staining in these two panels here compared to the healthy animals. Again, suggesting that neuturin increases the amount of mitochondria. Again, looking at the ability to use fuel. In the animals that had more neuturin, when they're provided with the glucose challenge, they're able to utilize the glucose and remove it from the system much more efficiently than your healthy animal. Again, supporting neuturin as a factor to improve the use of fuel. Also looking at the function of these animals. So when these animals overexpress neuturin, when these animals have, look at the top left, a slow neuromuscular junction structure, slow muscle fibers, more mitochondria, enhanced use of fuel, they have a much greater motor coordination and motor performance. I haven't shown the data here, but these mice also produce more force relative to their body weight, and they fatigue slower. So overall, enhancing this muscle or slow muscle phenotype using neuturin could potentially be a very potent way of delaying the disease progression, at least in the mouse model to start with. So the work that we're undertaking now is to actually test the hypothesis that if we overexpress this neuturin protein in a mouse model of sporadic MND, are we able to either prevent the disease from occurring or at least delay the progression of the disease? So we've started this work thanks through to funding from MNDRA, where we have been able to now inject a healthy mouse with a control AAV. So as Peter said, this is quite exciting because it's a clinically relevant approach that's being used in phase 1 studies of several other diseases. We've also given a control virus to the MND mouse that we're working with. And then we've given the virus to overexpress the neuturin protein in the MND mouse. And we need to wait a bit longer for the virus to get into the system and to do its work. But effectively, when this has occurred, we'll be looking at the quality of the neuromuscular junction. So similar images as what Peter had showed you. We're looking at the function of the mitochondria. so the energy producing aspect of the muscle. Look at the satellite cell number, because we know that in people with MND that these satellite cells are reduced, and also their ability to repair a perturbed. Look at the muscle size to see if we're able to prevent the severe atrophy in the MND mouse model. Naturally, we'll be looking at the amount of slow versus fast muscle fibers, and the slow should be fast, not fats, slow versus fast motor neurons, just to ensure that we have created this slow phenotype. And again, looking at their strength and their coordination, and also to see where this TDP43 protein resides. Are we causing it to stay in the place it's supposed to be to maintain that global health check, or is it still being shuttled and aggregated into parts of the muscle that where it should be, and that again impacts the connectivity between the motor nerve and the muscle. So we're quite excited by this work. It's very preliminary, it's very, very fundamental, but we hope to have some exciting results by the end of this year. So I'd like to give a big shout out to my team, very motivated team, a couple of postdoctoral researchers and 2 new PhD students, and also great collaborators that I've got overseas with the Neuturin viruses, and also some great collaborators that I work with here in Australia. And thank you very much for your time and your attention, and I'm happy to take questions.
Oh, thank you, Aaron and Peter. That was absolutely brilliant introduction to the world of muscles in motor neuron disease, because it's something probably we definitely haven't heard about in a series of talks, and I think perhaps is, I mean, I'm sure you'll agree, perhaps somewhat neglected sometimes as well. I'll fire in the question first, because it sort of jumped out at me. Is, I mean, these defects in the muscles, these are they driven from signals from the motor neurons? Do you know which way does it go? Should we rename motor neuron disease motor neuron and muscle disease, or what? I mean, sure, we can be as controversial as you want about that subject. Both, that's aimed at both of you, by the way.
Do you want to go first, Peter? Do you want to say yes, or will I say yes?
You can say yes.
Look, it's a really good question. It's something that the field's been grappling with. You know, are the defects in the muscle due to the feed forward, as you said, from the nerve to the neuromuscular junction, or is there some innate perturbation occurring within the muscle, or is it both? I think it may be a case of both. I don't know if the muscle causes, but it may be a very potent activator of the severity of the disease. And I don't think it's going to be 1 or 2 issues within the muscle. You know, I've mentioned 3, but there's another 10 or so that I could be talking about that are perturbed. And I think it's not going to be a magic bullet that fixes one particular perturbation. It's going to be several, and that's the complexity of this disease. It's not a TDP proteinopathy. It's not going to be a mitochondrial issue. It's going to be multiple factors. The hard part's going to determine which one, or if, occurs before the other, that causes a cascade within the muscle that then may push back up through the neuromuscular junction and cause that disconnect.
Yeah, I mean, I agree with Aaron. It's complex, and it's not like it'd be one thing or the other. We certainly know that that motor neurons are abnormal in motor neuron disease, and we also know that you can have early hyperactivity of upper and lower motor neurons, and we know that if you, for example, secrete too much neurotransmitter from the motor nerve ending to the muscle, that in itself can lead to the dispersal of these acetylcholine receptor patches. So we know there's a what we call a positive-negative balance between nerve muscle signaling. In other words, neurotransmitters can, if there's too much, can break down receptors. But what stops that is the AGRIN signaling pathway that will stabilize it. So it's kind of like a plus-negative system. And as Aaron said, the molecules that are existing in the muscle membrane that we look at, they are very much dependent on a lot of these other signaling mechanisms within muscle. So, for example, MuSK itself, which is this receptor that AGRIN can interact with, also can interact with muscle metabolism genes. So we know that this feedback's quite complex. One of the things that we want to do in our research is ask the question, if we take a motor neuron diseased motor neuron that carries an MND mutation and make it connect to a healthy muscle in culture, do we see normal clustering and specialization of receptors? But if we don't, that tells us that motor nerve also has this sort of feed-forward signaling. If it can cause the clustering receptors, even though it might carry an MND mutation and the muscle doesn't respond, then it kind of suggests that maybe there's a trigger in the muscle itself. So there's these sort of interplay. When we talk about the formation of nerve muscle connections in the animal or in the human, it's always a bidirectional signaling between motor nerves and muscle. So muscle will talk to nerve and nerve will talk to muscle. We know that, and that's been known for a long time. So there's a lot of other factors that get released by muscle, like growth factors that can maintain nerve muscle connections. We also know there are sticky molecules that are responsible for sticking the motor nerve to the surface of the muscle. And some of our other studies have shown that those sticky molecules are actually downregulated in MND. So we know that as well.
So do you ever see problems with the muscle either in patients or some of the animal models before you see problems in the motor neurons? Or is it possible to see that, to spot that first? So to kind of answer the question, I guess, you know, chicken and egg type theory, do you lose muscle before you lose nerve function?
That's the beauty of the animal models. You can actually go in before the animal develops the overt muscle weakness. And so that's the beauty of the animal models. You can go in before the motor neurons have died. You can even start to look at changes that Aaron's talked about in the muscle even before you lose those connections. So you can look at all those things before you see the sort of disconnect. For us, we know that the communication between motor nerves and muscle in one of the TDP-43 mice, the signaling mechanisms can be perturbed there, what we call the synaptic transmission. That occurs before we see any morphological or molecular changes. So we know these things can occur very early. Yeah. Yeah.
And just on that, in a couple of the mouse models that we've been working with, we see, for example, the mislocalization of the TDP-43 protein in muscle well before any change, any loss of body weight or loss of coordination occurs. We see impairments in mitochondrial function as well before we see changes in body weight, changes in muscle force production. So there is something going on early, and that's observed also in the SOD model.
So, so from a patient perspective, does exercise help? You know, can you build up your muscles, or I guess in extreme cases, could you take anabolic drugs to give your muscles a massive stimulus to help?
That's look, I just saw that question come through, and that's a great question, and it's a question that I'll have to be brutally honest. I'm an exercise physiologist in a previous life, and we've done some exercise studies with mouse models. And if you look at the exercise literature in the MND field, in mouse models, I'll start with, it hasn't been done as well or as frequently as it probably should have. So the answer there is it doesn't seem to have a negative impact on the muscles, on the animals. It may delay in some of the mouse models, but my concern is the lack of reproducibility in that particular field. With respect to studies with humans, it's quite controversial as well, because there hasn't been enough research done yet for a couple of reasons. The difficulty in getting a sufficient number of people to complete the exercise programs, and also with a significant number of people with the similar disease onset, disease stage. So it really requires a lot more research to be done. There is a study going on at Victoria University at the moment where they're doing an exercise study. I think it's a quite interesting study that they're saying to undertake. My opinion is that while someone can maintain physical activity, it would be beneficial than not, because physical activity is important in muscle function and muscle neuromuscular junction connectivity. But in terms of a specific training regime to try and delay, the research hasn't been done yet. Sufficient amount of research hasn't been done for various reasons.
And what I think is really interesting is that, and this goes to what Aaron was talking about in his talk about this idea of this muscles, the slow type muscle contractions. When we look at the patient muscle biopsies very early on in disease, before they develop a lot of muscle weakness, muscle fiber conversion is about 50%. So we're seeing 50% more slow type fibers, and they're losing their fast twitch fibers. And so when you give that to a pathologist and they look at that, their immediate diagnosis is that the patient is undergoing denervation, reinnervation events, and that the motor neurons that are being lost are the fast motor neurons, the ones that connect to the fast twitch muscle fibers. And so when those muscle fibers lose their connections, they're reconnected by the slow type motor neurons. So they're converting the fast to the slow. So we already know that the muscle is trying to maintain connections, and whatever neurons that are left, that maybe the slows, have the ability to connect and then basically turn them into a slow type. The interesting thing for Aaron and I is that over time, we'll also lose those connections. So how do we keep those connections as well?
So is there a pharmacological way to try and overcome that? I mean, obviously you see bodybuilders, etc., and Ben Johnson, etc., who pump themselves full of drugs to get stronger and faster. Is there any correlation with MND?
With respect to any of the fiber type conversions, no, those anabolic factors won't be influencing or maintaining a slow fiber conversion. I'm not sure if that was the question. In terms of taking anabolic steroids, you can't just take an anabolic steroid and sit on the couch and do nothing. An anabolic steroid doesn't make you get bigger muscles. You need to be doing the work. I'm not condoning taking anabolic steroids at all, but the people who do take anabolic steroids actually train a lot, like more so. It assists with recovery and muscle growth because you can do more. So it's not just an injectable thing.
And I guess perhaps like many potential treatments in MND, do you think if you do find a way to stimulate the muscles, then the earlier you can do it in disease, the better?
Yeah, I mean, I think the biggest problem in any sort of neurodegenerative disease sphere is biomarkers. Can you find a biomarker that is a predictor of the disease before you get overt symptoms? And so certainly in the muscle, the molecules that Aaron and I have been talking about, they may actually be biomarkers. I think the exciting thing is to start, this is the advantage of looking in the animal models, whether some of these markers are coming up really, really early. And then if they are they coming up really, really early in the humans? The problem is that people are really only coming to the clinic when they've got symptoms. You'll always have a subgroup of MND patients that are genetically linked, and the theory there is you might be able to start looking at those patients before they get symptoms. But you're talking about 80 or 90% of the population that has this terrible disease. There's not a biomarker and there's no genetic link. So you really can't pick them up until they come into the clinic. When you talk to the clinicians now, I think the clinician's diagnosis of this terrible disease is much, much better. And so in the old days, a patient would have to go through so many doctors and clinicians before the final diagnosis was made. And so diagnosis has been made a lot quicker these days. So that's good. But yeah, there is a desperate need for biomarkers, and there are a lot of researchers looking into biomarkers.
So just on that, with respect to the muscle, especially it's hard to identify something in the muscle itself at this point in time. What we're looking at is with the models that we've got, because we can induce and control when the disease starts, and in these animals, taking a large number of tissue samples and blood and plasma samples very, very early on, before there are any functional changes, to see if there are any relationships with changes inside the muscle, several of those I've already mentioned and others, and do they correlate with any changes in proteins within the plasma, within the serum, different types of proteins, different types of RNAs, to see if we can develop a potential signature that may be linked to the muscle, because there are factors within the muscle that are secreted when there is sick muscle and they shouldn't be in circulation. So we're looking at it that way.
Cool, thank you both. So we're pretty much out of time now, so I'll just wrap up. So look, those were fascinating talks, and I really think our funds are being extremely well spent, because we're really looking forward to seeing how those potential therapies develop in both your hands. For those or everyone watching tonight, you all get a link to our recording, so please feel free to distribute that across your networks as well. I think these stories need to be told as widely as possible. It's great to hear about this aspect of MND. If you're interested in previous editions of State of Play, then go to our page on MNDAustralia.org research and look under the news and updates. Also on there, you'll find a series of monthly research updates we just started, called Research Directions. So that just gives you a bit of a snapshot of what's happened in the last month in the MND research world. That's all for tonight. Thank you again to Peter and Aaron. I really enjoyed that talk, and I reckon everyone else would have too. It's like I said, really good insight into an aspect we don't hear enough about. So thank you to everyone for dialing in, and good night, and we'll see you for the next State of Play. Thank you all.
Thank you. Thanks for the opportunity.
And thank you very much for those that took the time out to listen to Aaron and myself. I very much appreciate it.
Me too, Pete. Thanks very much, everyone.
Good night, everyone.